process of the biological system. So for the past topics natin, we have discussed already yung different catabolic pathways from glycolysis, fatty acid oxidation, amino acid degradation or oxidation, and other phosphorylation methods, which includes which involves production of energy through ATP and um, proton carrier NADH or NADPH. So those are our anabolic pathways. And at this point, we will be discussing na yung that's our catabolic pathways, and now we will be discussing anabolic pathways, which will now use up yung ATP and NADPH and NADH in order to produce precursors or biomolecules, which will require in different reasons. Okay. Si catabolism, we will that's usually the oxidative process. Si anabolism naman yung ating reductive process. Okay? So catabolism and anabolism nangyayari siya simultaneously in the biological system, which create and maintain balance in the living cell. So if one side lang yung nangyayari, that can be a great problem. Kasi if catabolism lang will be the primary biological metabolism that will occur in sa body, edi puro energy lang tayo. Hindi nagagamit yung energy na nagagawa. So anabolism and catabolism are parang two sides of orderliness in the biological system. Okay? We will be discussing particularly this afternoon yung carbohydrates biosynthesis. So I have to recall na primary way of our biological uh, degradation of our carbohydrates from glucose from uh, polysaccharides that we have been consuming through our meals has been degraded to form small units of pyruvate na e-enter sila to sa Krebs cycle to give off energy by oxidation. So pabalik naman tayo on production naman tayo of those sugars. Okay? So sa carbohydrates biosynthesis, we will be focusing in plants. Bakit? Kasi plants have a great versatility in handling carbohydrates. So plants are autotrophs. So they can only produce, or they digest or they consume inorganic components like carbon dioxide to produce organic compounds in their system. Okay? So also biosynthesis occurs primarily in plastids, which is a very unique which help dun sa movement ng mga intermediates or ng mga metabolites within the cellular compartments no plant. Okay, so that plastids specifically, we will be discussing different kind of plastids uh, that the plants have. Familiar kayo siguro, familiar with chlorophyll, since elementary naman ata inaalala yung chlorophyll, which attracts light. So ayun, we will be discussing about it more in detail. So chlorophyll, example of plastid, is a very unique characteristic of the photosynthetic organisms, specifically plants, that help them to synthesize different precursors na kailangan ng nahulog po. sorry. Ayan. Which needed, sorry for that, ng plants for it to grow, for it to live, and for it to propagate. Okay? Another reason kung bakit it can be said that A plant have a great versatility on carbohydrate synthesis and consumption is plants have sufficient metabolic flexibility in order to adapt in changing condition. Given that plants are there are not motile, so hindi nila kayang lumipat na kung saan-saang lugar, they have to be adaptive in their environment wherever sila naka-root or wherever sila rooted. So yung they have to be metabolically flexible in order for them to live on that whatever kind of condition or environmental condition. Okay? And lastly is the production of the thick cell walls of plants significantly contributes to the proportion of plant cell carbohydrates. So one of the process of plants in handling carbohydrates is formation of cell walls. So the cell walls, we already know that it is only available for plant, which is uh, thick part of the cells, which we will explain later kung bakit siya mahalaga sa plants. Pero one of the reasons is for the control of water absorption and water secretion. Okay? Now we will be discussing on the photosynthetic carbohydrate synthesis. So photosynthetic means we are initiated by the light. So kailangan natin ng light in order for this metabolic process proceed. Plants and photosynthetic microorganism utilize carbon dioxide and water for the synthesis of carbohydrate. So we have learned naman noon na, di ba, between the relationship of man and the plant, yung nag-give off, nag, 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 nag-inhale tayo ng oxygen which is released by the plant while yung release natin na carbon dioxide is used up naman by the uh, plant to to live. Ayan. So it reduces carbon dioxide, a really highly oxidized carbon, at the expense of energy and reduces power furnished by the ATP and NADPH. So this is what we say in anabolic process, whereas yung carbon dioxide natin will be converted to reduced form in while using ATP, so energy consuming siya, and NADPH, which contains proton to help or assist in production of these precursors that are generated by light-dependent reactions of photosynthesis. So specifically in plants, we will be using this ATP and NADPH as uh, activators to proceed yung ating reactions or yung ating biosynthesis. Carbon dioxide is the sole source of carbon atoms required for the biosynthesis of cellulose and starch, lipids, and proteins, and the many other organic components of plant cells. Okay? So carbon dioxide lang yung way of the plant to produce yung ating mga biomolecules. Specifically for this topic, yung carbohydrates, and since doon lang possible manggaling yung you call it yung carbon, essential din siya in production of different metabolites. Metabolites, yung secondary metabolites, hindi siya yung mga biomolecules na protein, na lipids or carbohydrates, but other organic components in plants which are sometimes considered as biologically active compounds. Yun yung mga components ng mga halaman that gives off coloring, gives off scent, gives off biological importance, like pwede siyang gamitin for as anti-diseases or for any illnesses. So yun. So essentially carbon dioxide lang yung pwedeng panggalingan ng mga carbons sa mga compound. Okay? Bukod dun sa mga carbons na existing na dun sa loob ng plants. Okay? So we will be tackling about most of this. We'll be tackling about the carbon dioxide assimilation. So what's with carbon dioxide assimilation? So it occurs in chloroplast. So yung chloroplast, yung green pigment or green organelle of the plant, a unique enzymatic process that converts carbon dioxide to simple reduced organic compounds. So, yung carbon dioxide, once absorbed by the plant, it will be now fixed, nagkakaroon ng fixation, and it will undergo different chemical reaction in order to sustain the needs of the plant in terms of the carbohydrate and other precursors. So it is called also carbon dioxide fixation or carbon fixation. So specifically, Calvin cycle, yung isa nating pag-usapan of na way of carbon fixation, is also called photosynthetic carbon reduction cycle. The cyclic pathway of carbon dioxide assimilation. So this is one of the cyclic pathway for the carbon dioxide assimilation. So it was elucidated in early 1950s by Melvin Calvin and his colleagues. So pinangalan kay Melvin Calvin yung na-discover na biosynthetic pathway. So this is the gist, or this is the general, this is the Calvin cycle itself, pero we will discuss it in details in mga next slides. So as you can see in this chloroplast containing your stroma, which captures the light, ayan, si carbon dioxide will enter the Calvin cycle in form of uh, phosphorylated six carbon six membered carbon using specific enzyme which helps it to be converted at maging organic compound, and so on and so forth for the Calvin cycle in order to produce your glucose, yeah, and other sugars. Okay? So ang endpoint of our explain natin ito. Okay. At the endpoint of our Calvin cycle is the production of glyceraldehyde triphosphate, which will lead to production of sugars. Okay? So para siyang Krebs cycle in just production of energy. So plastids are organelles unique to plant cells and algae. So there are non-plant organism that has photosynthetic approaches then in order for them to live. May mga bacteria that uses photosynthetic for that. Pero for this moment muna, we will be discussing about plant. Plants have unique reaction sequence for reduction of carbon dioxide to triose phosphate and the associated reductive pentose phosphate pathway. So yun. We have discussed already yung PPP, pentose phosphate pathway, which uses pentose sugars. And in this case, kay plants, it uses this triose phosphate and convert it to pentose phosphate, and it to produce the specific carbohydrates. Okay? So we will discuss it in details, and we have to be reminded that still, since ito ay metabolic pathway, kahit na siya ay anabolism, it can also be regulated. So regulated by specifically on different enzymes. One example, four here that have been included is the reduction of the disulfide bonds by electrons flowing from the photosystem 1, which will be discussed later. Change in pH, so there is a specific effect ng microenvironment of the uh, plant system. So specifically on the pH level or the acidity or the basicity of your plant system, and the magnesium 2 plus ion concentration, it can be affected sa illumination. So we will understand later ano yung ibig sabihin na. Conventional allosteric regulation by one or more metabolic intermediates. So ibig sabihin, one of the products or one of the intermediates within the system or within the reaction can allosterically regulate our one of the enzymes involved in the reaction to stop or to induce the production of your specific target compounds. Covalent modification or phosphorylation, so that's how we do to activate such compounds, or how can we activate, or for activation ang phosphorylation. Pero certain reaction in biological system can be deactivated naman once you remove yung phosphorylation, phosphyl group. Okay? These are the examples of your plastids. So plastids, they are self-producing organelles bounded by a double membrane or containing a small genome that encodes some of their proteins. So plastids, you can treat it as parang uh, yun as a cell-like organelle kasi it can produce itself by its own by is the sites for most synthetic activity in plants. Okay, so example of it is chloroplast, which is the sites for the carbon dioxide assimilation. The enzymes for this process are contained in the stroma. So, well, that's figure here, yung mga nasa loob dito. The soluble phase bounded by the inner chloroplast membrane. So, chloroplast actually dominates usually the plants. Another example of plastids is amyloplast. It is a colorless plastids and have no internal membranes analogous to the thylakoids of the chloroplast. Thylakoids yung part of the stroma, which it dominates naman yung mga non-photosynthetic tissues that store in large quantities. So this is usually can be seen on tubers dun sa mga halaman or mga plant species na hindi na expose sa light. Okay? So amyloplasts ay they are colorless because they lack chlorophyll and other pigments found in chloroplasts. Another example of plastids is proplastids, which is formed by conversion of chloroplast by the loss of their internal membranes and chlorophyll, which is also interconvertible to amyloblast. So this is your proplastids, which is from chloroplast, and this proplastid can be converted to amyloblast. Okay? The relative abundance of the plastid types depend on the types of plant tissue or on the intensity of illumination. So of course, you will yung plant, it will adapt to make or make to make the chloroplast available to the area na kung saan alam niya na or uh, alam niya na it will make more efficient absorption of the light. So usually kaya yung mga chlorophyll can be seen sa leaves, syempre sila yung mga mostly exposed in the light. The inner membranes of all types of plastids are impermeable to polar and charged molecules. So traffic across this membrane is mediated by sets of specific transporters. Since this plastids is not prone for permeation of polar or charged molecules, unlike the cells in the cells in our body. So may certain transporters or mga shuttles that can help or assist yung pag-enter and paglabas ng mga inorganic or organic molecules which helps para ma-continue yung reaction within the system. Okay? Right. So we will be discussing yung carbon dioxide assimilation. So what happens with the Calvin cycle. So fixation of carbon dioxide into 3-phosphoglycerate is the first stage in our Calvin cycle. So what happens here is that 3-phosphoglycerate is produced by fixation of your carbon dioxide in the help of your specific enzyme. So 3-phosphoglycerate was proven to be one of the first photosynthetic intermediates by isotope labeling with radioactive carbon dioxide 14. So Well, familiar kayo dun sa isotope labeling, whereas pinagre-react is ang isang isotope labeled component with an existing uh, biomolecule, and minomonitor on where mo makikita yung isotope na yun after the reaction. The same with this, they have introduced this for carbon dioxide 14 uh, sa algae. Ay, kasi ako. Okay. The many plants in which this three-carbon compound is the first intermediate are called C3 plants. Okay, so you have to be familiar with this kind of ano yung mga kind of plants that uses this kind of this fixation. So yung una makita is C3 plants if gumagamit ng three carbon containing uh, compound yung cycle, it is called C3 plants. Kaya ko to sinasabi na you have to be concerned with these C3 plants, C4 plants, C2 plants. Kasi most of this name or uh, yeah, most of these topics ay lumabas nung boards namin. And well, unluckily, hindi kami familiar with the terms or siguro nalimutan na namin siya. Kaya we somehow nagkakaisa kami mga magkaklase na hindi namin nasagutan ng maayos yung biochem part nung exam namin that time. So yun. So C3 plants, C4 plants, yung mga ganong wordings usually appeared sa board exam. Okay? So for this case, we will be dealing with the C3 plants, which uses three phos which uses three carbon containing compound. So what happens? So sa stage 1 natin, ribulose 1,5 bisphosphate carboxylase oxygenase, yung main enzyme natin which assists yung carbon dioxide to enter our Calvin cycle. So in short, yung Robisco catalyze incorporation of carbon dioxide into an organic form, catalyzes the covalent attachment of carbon dioxide to the 5-carbon sugar ribulose excuse me, 1,5-bisphosphate, which leads to formation of unstable 6-carbon intermediate, which is cleaved naman later on to produce 3-phosphoglycerine. This plant enzyme has low turnover number. So ito yung itsura niya. This enzyme has ano ba dito? Yun siya na ilagay. So meron siyang 8 similar sites. Meron siyang ano tawag ba? Ano tawag ba? Yan, subunits. Meron siyang 8 similar subunits, yung Robisco mo. And those are active sites for the incorporation of carbon dioxide to your organic component in cells. Okay, so this plant enzyme has low turnover number, so only 3 molecules of carbon dioxide per seconds and molecule of Robisco at 25 degrees Celsius. So medyo mababa yung production or yung fixation of your carbon dioxide in every unit of Robisco. So ano yung ginagawa ng plant in order to overcome this? So they produce large amounts of this enzyme. So yung Robisco can be kapag tinaray mo siyang i-extract in your plant sample, they say na parang almost 50% or half of those sample contains Robisco. So that is the way of plants in order to overcome or to supply the needs of the plants for the carbon containing compounds. Okay? So central to the proposed mechanism for plant Robisco is that the carbamylated lysine residue with a bound magnesium 2 plus ion. So as we have learned sa mga nakaraang slides, mga nakaraang topics, that magnesium or mga metal natin are good chelating agents, and they are part of the mostly part of the system in order to assess yung formation of compounds. We have seen also already si magnesium 2 plus sa ATP in order to neutralize yung negative charge property of your ATP. In this case, magnesium 2 plus ion assists in orientation and attachment of your reactants with the active site. And here, mention also that it helps to polarize your carbon dioxide, making it more electrophilic to be attacked by your nucleophilic ribulose 1,5 bisphosphate. Okay? Opening it, polarizing carbon dioxide helps it to be open for nucleophilic attack by the 5-carbon enodiolate reaction intermediate formed on the enzyme. So the resulting 6-carbon intermediate breaks down to yield 2 molecules of 3-phosphoglycerine. Okay, so this is what happens in your Robisco, sorry, in your Robisco. So, yung Robisco mo, meron siyang acid residue, and one of the important part here is your carbohydrate lysine side chain. Okay? So, and in your magnesium 2 plus. What happens here is that yung ribulose mo forms an enodiolate at the active site. So, the carbohydrate lysine side chain will attack your hydrogen here in your ribulose phosphate, forming your enol. Yeah. And afterwards, this alkene group being formation of another carbonyl group by the histidine residue nearby it, attacking your hydrogen, giving off your bond to oxygen and carbon. And electron here will attack your now more electrophilic carbon dioxide by the purpose of your magnesium 2 plus. So, mas prone na siya for electrophilic attack. Now, attach na yung carbon dioxide mo to your organic compound. So, carbon dioxide polarized by the proximity of magnesium 2 plus ion undergoes nucleophilic attack by indolate, generating a branch 6-carbon sugar. So you have now your keto acid intermediate. Hydroxylation occurs at the C3 here. So ito yung next na part. So what happens here is that you have now hydroxylation. So hydroxylation happens here, movement of your electron, and giving of your bond here to form your carbon anion. And now you have your one of the 3-phosphoglycerate. So yung isa mong 3-phosphoglycerate is from your ribulose 1,5 bisphosphate, and your another phosphoglycerate can be formed here by your carbon anion and production of your phosphoglycerate, which contains yung carbon dioxide mo from the outside, yung fixated na carbon dioxide. Now you have your carbon ion, protonation generates second molecule of 3-phosphoglycerate. Ayan. So now you have 2 units of your 3-phosphoglycerate. Okay? Ayan. So that's what happens sa formation of your 3-phosphoglycerate, attachment of only 1 unit of carbon dioxide. Okay? So regulation of Ribisco can be done since it is the prime target of the regulation. Since ito yung first part of the Calvin cycle, it can be a good part for the regulation or the control of the biological system. It is inactive until carbamylated of the epsilon amino group of lysine 201. So as you can see here, yung NH3 is an inactive, this rubisco and 1,5-bisphosphate na ribulose is inactive since you have no reactive site for your lysine. So ribisco with unmodified lysine 201 and bound ribulose 1,5-bisphosphate is inactive. Uh, ang ginagawa ni ribisco in order to remove yung ribulose mo, to help na ma-uncover yung lysine and ma-activate siya is by introduction of ATP. So you use up energy using enzyme pyruvisco activase to give off your ribulose 1,5-phosphate. Now you have your free lysine 201 residue, which is now prone for attack of a carbon dioxide. So epsilon amino group of lysine is carbomoylated by carbon dioxide, and magnesium 2 plus binds to the carbamoyl lysine, activating rubisco. Now this activated rubisco can be used for the production of your, or for the capture of your carbon dioxide and production of your 3-phosphoglycerine. So yun yung idea between the rubisco being active and inactive. So that's how we can regulate our rubisco, right? So after formation of your 3-phosphoglycerate, it can be now converted to glyceraldehyde 3-phosphate. So conversion was done by reversal reaction steps in glycolysis that uses, kung familiar kayo, kung natatanda nyo pa siya, yung glycolysis uses NADH for the conversion for this specific compound. But in plants, we will be using NADPH instead as the nucleotide cofactor. So the chloroplast stroma contains all the glycolytic enzymes except phosphoglycerate mutase. The stromal and cytosolic enzymes are isozymes. Yeah. This is just discussing on what kind of enzymes are involved in your conversion. Okay. So what happens here is that conversion was done by the reversal reaction steps in glycolysis. So here, your 3-phosphoglycerate and will be converted to 1,3-bisphosphate glycerate in assist of your kinase. So you will use up again in an ATP. And now your 1,3-bisphosphate glycerate, ayan pala, meron pala ito. So the stromal 3-phosphoglycerate kinase catalyzes the transfer of phosphoryl group from ATP to 3-phosphoglycerate, yielding 1,3-bisphosphoglycerate. So yung 1,3-bisphosphoglycerate na nandito. Okay? Afterwards, an ADPH and a proton will be donated to your 1,3-bisphosphoglycerate in order to convert it to your glyceraldehyde 3-phosphate. Okay? And ADPH donates electrons in a reduction catalyzed by the chloroplast specific isozyme by glyceraldehyde 3-phosphate dehydrogenase, producing your inorganic phosphate and NAD+. Right? So this is the ito yung itsura niya. Okay? So you have your 1,3-bisphosphate glycerate. Upon addition of NADPH and H+, it is now reduced to have your glyceraldehyde 3-phosphate. Okay? Right? High concentration of NADPH and ATP allow this thermodynamically unfavorable pair of reactions to proceed in the direction of 1,3-bisphosphoglycerate. So as we have mentioned earlier, that ATP is used for activation of specific compound, and this assists for the further reaction of these compounds. Triose phosphate isomerates then interconverts glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So familiar na kayo dito, si G3P and DHAP is now converted. So since Si dihydroxyacetone can exit your cytosol using this inorganic phosphate triose phosphate anti-porter. Now it can lead to either production of your fructose 1,6 phosphate or production again of glyceraldehyde phosphate to produce ATP in form of glycolysis or formation of sucrose as your storage. Right? Another way then of the preparation of or another way to store your carbon dioxide fixated organic compound is to production of your starch. So we will discuss it later then. Pero yung flow niya is that your glyceraldehyde phosphate will be uh, coupled with your dihydroacetone phosphate using aldolase to produce your fructose 1,6 phosphate within the cytosol. And this fructose 1,6 bisphosphate can be converted or can be uh, phosphatase. So, mariremove yung phosphyl group mo to prepare your fructose 6 phosphate, and later on, it can be used up to create starch. So, starch and sucrose are the main storage system for the carbohydrates in plants. Okay. Most of the triose phosphate that's produced is used to regenerate ribulose 1,6 stage 2 pa rin to. Yeah. Yeah. So, ayun, most daw of the triose phosphate, as you can see earlier, since meron tayong na-produce na dalawang triose, we are using it up then to regenerate yung ribulose 1,5 bisphosphate, which is initially used for the incorporation of the carbon dioxide. The rest is either converted to starch in the chloroplast and stored for later use, or immediately exported to the cytosol and converted to sucrose for transport to growing regions of the plants, and, for the development and growth of your plants in its leaves, in its fruits, so on and so forth. Okay? And can be degraded by glycol. Nadoble ko pala siya. Okay. So, ito yung medyo summary of the fate of nung triose phosphate. It can either be regenerated to ribulose 1,5 bisphosphate. It can be converted to starch for later use, or it can be used up by it can be converted to sucrose for use up of energy, and for another storage, and it can be degraded by glycolysis to provide energy. So that can be this is the 4 fates of your triose phosphate. Depende sa kung ano yung kailangan ng plant mo in that specific time or specific condition. Okay? Excuse me. Stage 3 for the carbon dioxide assimilation is the regeneration of ribulose 1,5 bisphosphate from triose phosphate. So medyo marami siyang reaction. It involves 9 different reaction from coupling, from breaking it up, from another coupling, then another coupling, conversion, in order to produce your ribulose 1,5 bisphosphate. So what R15B5 BP or ribulose 1,5 bisphosphate should be constantly regenerated for carbon dioxide assimilation to follow. The same with ano nga yun sa Krebs cycle, oxaloacetate. It should be regenerated kaya siya in cyclic form in order for the reaction proceed. Dito rin, ribulose 1,5 bisphosphate, organic compound which is help to incorporate carbon dioxide, inorganic carbon dioxide in your to convert it to organic compound. So kailangan siyang ma-regenerate. So what happens is that glyceraldehyde 3-phosphate that has been synthesized or produced by your stage 1 and stage 2 of carbon dioxide assimilation is coupled with dihydroacetone phosphate. So this reversible condensation of glyceraldehyde 3-phosphate with dihydroacetone phosphate to yield your fructose 1,6 bisphosphate. So it uses an enzyme aldolase. Excuse me. Pati na ilong to. Ayan. So, yung fructose 1,6 bisphosphate mo naman will be cleaved excre- uh, expelling inorganic phosphate. So cleavage of fructose 1,6 phosphate forming fructose 1,6 phosphate and inorganic phosphate using the enzyme fructose 1,6 bisphosphatase. So phosphatase, nagtatanggal lang siya ng phosphate group. So this is an strongly exergenic and essentially irreversible manner. So yung makikita nyo naman dyan kung ano yung mga reaction which are irreversible, yung mga single-headed arrow and naka-color blue yung kanilang color. So those are the irreversible reaction. So as you can see here, some of the irreversible reaction involves attachment of ATP. So those are kasi activated compound na yung dalawang containing or ribulose 1,5-phosphate. Okay? Next step will be the transfer of your 2-carbon ketyl group. So nagredundant. This is ketyl group. So here, nasan tayo? Ayan. So yung fructose 6-phosphate mo, which has been removed from your, which has been produced from your fructose 1,6 bisphosphate, is now will receive a ketyl group using transketolase to form to form yung glyceraldehyde mo, acylulose 5 phosphate. So naka-receive siya ng 2 carbon containing ketyl group, and yung fructose 6 phosphate mo naman will be now erythrose 4 phosphate. Ayan. So transfer of 2 carbon ketyl group of fructose 6 phosphate to aldose phosphate acceptor glyceraldehyde 3 phosphate, forming the pentose cellulose 5 phosphate and tetrose erythrose 4 phosphate. Ayan. So you now have a 4-membered carbohydrate and 5-member carbohydrate. So I hope familiar pa kayo dun sa mga carbohydrates natin. Yung wait lang. Sinong biochem 1 nyo? Okay, good. Si Ma'am Joy. Si Ma'am Joy yung nag-introduce sa amin sa biochem din. And she made us be familiar with different hexoses. Ginagamit niyo ba yung all-altruths? All-altruths, making gums, parang ganun. So, naturo sa amin yun. And I hope familiar pa rin kayo dun. So, the enzyme that is being used, wait lang, excuse me. Tapos na ata si Nene, nag-chat sa akin. Ewan ko lang kung nagpapasundo sila. Hindi, nagkaklase ako. Sabihin mo, mag-chat na lang. Ay, chat nyo si Nene. Chat mo na si Nene. Alin? Pwede naman kay Arnie, kay Abby. So yun, kahit sabihin ko naman siya. Okay. Sorry. Okay. The fructose 6-phosphate to aldose phosphate receptor. Yan. Now you have your 4 carbon-membered carbohydrate and 5-membered carbohydrate. So meron ka na ditong pentose, meron ka naman ditong tetrose. Okay? So this tetrose will now be reacted with different with another DHAP. Ano mahalaga dito? Yeah. Dito sa transketolase mo, well, you just transfer a ketyl group. Kailangan ng dito is your TPP as your prosthetic group, which requires also magnesium 2+. So as you can see, magnesium 2+ ion is a very important component of the biological system. for different types of reaction. Okay? The next step will be the combination of your erythrose. Ayan. Combination of your erythrose 4-phosphate with another dihydroxyacetone phosphate in production of your 7 carbon containing carbohydrate, or your pseudoheptulose 1,7 biphosphate, bisphosphate. Okay? So combination of erythrose using another aldolase. So the same manner siya with your step 1, which gumamit ka rin ng aldolase. So pag aldolase, you just combine different compound. Okay? So here, ito, pinakita dito yung nangyari sa ketolase. Dito naman, conversion of your, yan, dito, okay. So dito, this is the general manner on what happened for the ketolase. So meron kang aldose acceptor, which is your glyceraldehyde kanina, and your ketose donor, which is your phosphate or your fructose phosphate. So nagkakaroon lang ng transfer of your ketol group. So for example, yung fructose 6-phosphate mo with glyceraldehyde, yung N-carbon keto group mo will be transferred to your glyceraldehyde phosphate. Excuse me. Right. Ano naman meron dito? Yeah, this is another transfer then of carbon which will happen naman later. Combination of erythrose to produce your 7 carbon containing carbohydrate. And now you have your conversion of pseudoheptulose 1,7 bisphosphate to pseudoheptulose 7 phosphate. So you will be using here phosphatate. Gumamit ka naman ng phosphatate. Your pseudoheptulose 1,7 bisphosphate using your bisphosphatase will be converted to pseudoheptulose 7-phosphate, yielding again inorganic phosphate. Okay, so this is another irreversible reaction. So your 7 carbon-containing group naman, what will happen to it is magta-transfer ulit ng keto group from pseudoheptulose to glyceraldehyde for you to form 2-pentose phosphate. So ito yung kanina. Okay. Sorry. Allergy ata ako. Pseudoheptulose, you have here, yung 7 containing compound, will, yung yung keto group mo dito will be transferred to another glyceraldehyde phosphate, which is a good acceptor or aldose acceptor, to have your ribose 5-phosphate and cellulose 5-phosphate. So, meron tayo ulit cellulose 5-phosphate and another one is your ribose 5-phosphate. Okay, so this is what happens in your conversion of pseudoheptulose to cellulose 5-phosphate. Okay, we have mentioned earlier na gumagamit tayo ng TPP or thiamine pyrophosphate as your cofactor to assist in the production of in the transfer of your ketol group. So pseudoheptulose 7-phosphate or ketose donor will be attached to your TPP and free of your ribose 5-phosphate. Your keto group naman, this is a carbon ion stabilized by resonance, will now be attached to your glyceraldehyde 3-phosphate to produce your cellulose 5-phosphate. 2-carbon fragment is temporarily carried on transketolase cofactor TPP and condensed with the 3 carbons of 3-glyceraldehyde 3-phosphate. Okay. Alright. Afterwards, meron ka ng cellulose 5-phosphate and ribose 5-phosphate. What happens now? So yung ribose 5-phosphate mo will be isomerase to form your ribulose 5-phosphate. The pentose phosphates formed in the transketolase reactions, yung ribose 5-phosphate mo and cellulose 5-phosphate, are converted to ribulose 5-phosphate. Yung kailangan natin for the starter of your Calvin cycle. So here, yung ribulose 5-phosphate mo uh, will be formed by isomerase here. Isomerase here. Ito naman is epimerase. Ay, excuse me. Okay. Afterwards, yung ribulose 5-phosphate mo naman dito will be converted to ribulose 1,5 bisphosphate by attachment of another ATP in order for you to produce your ribulose 1,5 bisphosphate using your ribulose 5-phosphate kinase. So kinase, nag-add ka lang na phosphoryl group. Okay? So that is how we accomplish in production of ribulose 1,5-phosphate, our 5 carbon containing carbohydrate from our produced glyceraldehyde na incorporated with carbon dioxide. Okay? Yeah. So yeah. Yun na naman siya. You started with your three carbon containing compound to produce your 5 carbon containing compound with two phosphate group and convert it to one phosphate group containing six-membered. This is six-membered pala, not five. Six-membered carbohydrate. And upon transfer of its ketol group, you have your 5-member and and 4-member carbohydrate and converted to 7-membered, 7-carbon-membered carbohydrate with dihydroacetone phosphate, and so on and so forth. It will be converted and another transfer of ketol group to produce your 5-membered carbon carbohydrate, and it now can be converted to ribulose 1,5-phosphate. Okay, so that's the final step of our Calvin cycle. Okay, reduction. Ay, sorry. Ito lang yung process, stage 3 lang. This is the summary of your stage 3 pala. Yeah. This is the final step for the Calvin cycle in order to regulate at reproduce your ribulose 1,5-phosphate. Uy, may class ako.
May kala mo pa man din dito.
Uy, ma. Okay. For the summary of your Calvin cycle, ito siya. So you have your starting material, your carbon dioxide and water, which will enter your Calvin cycle in assist of your rubisco to form your 3-phosphoglycerate. Then your 3-phosphoglycerate converted to glyceraldehyde 3-phosphate. Ito yung iyong stage 1. Wait lang. Ayan. Will be converted to glyceraldehyde 3-phosphate, interconvertible to dihydroxyacetone phosphate, and can be converted to glyceraldehyde 3-phosphate, which will undergo to different reaction in either glycolysis or formation of storage of carbohydrates. And to reproduce your ribulose 5-phosphate for your stage 3, you will be using up yung 5, excuse me, nag-allergy ako at kate. Ito. Ayan. your glyceraldehyde phosphate and dihydroacetone phosphate will be converted to your ribulose 1,5 phosphate. Okay? So the net result of 3 turns of Calvin cycle is the conversion of 3 molecules of carbon dioxide and 1 molecule of phosphate to a molecule of triose phosphate. Bakit tatlo yung ginagamit natin? Kasi we have mentioned earlier na tatlo lang yung kayang i-accommodate ng Rubisco for uh, every mole of Rubisco. Tatlong carbon dioxide lang yung kaya niyang i-welcome. Okay? So, ayan. Excuse me, wait lang. Okay? So, yung tatlong carbon dioxide mo and water will help to produce yung 3-phosphoglycerate. Remember na from 1,5 bisphosphate to. So, 5-membered ring plus 1, so 6. Unstable yon, so it will split up to 2 units of 3 carbon-containing carbohydrate. So, yung 3-phosphoglycerate. So, times 2 ng 3 mo, so 6. Now, you will use up 6 ATP, formation of 6 ATP to form your 6,1,3-bisphosphate glycerate, 6 glyceraldehyde. One of the produced glyceraldehyde will be ejected to, sensitive na ako, will be subjected to different reaction sa biological system, and the other 5 will be used or will be used in order naman ma-restore yung ribulose 1,5 bisphosphate mo, which will react ulit sa another carbon dioxide unit. So in NADPH and ATP are produced in the light-dependent reaction of photosynthesis in about the same ratio as they are consumed in Calvin cycle. So, di ba yung worry natin in our biological system is how much energy yung may-use up and how much energy yung mapaproduce. And fortunately, in plants and light-dependent reaction, nagkakaroon naman ng balance in the usage of energy and your NADPH para magpatuloy yung reaction. Okay? So 9 ATP molecules are converted to ATP and phosphate in the generation of a molecule of triose phosphate, and 8 of the phosphates are released as inorganic phosphate and combined with 8 ADP to regenerate ATP. So this later will be converted naman, yung ADP mo will be converted to ATP para ma-regenerate naman yung ATP. Excuse me. Okay, the 9-phosphate is incorporated in the triose phosphate itself to convert the 9th ADP to ATP. A molecule of inorganic phosphate must be imported from the cytosol. Yeah. So this is just the summary on what happens in your Calvin cycle in plants inside the chloroplast in production of the glyceraldehyde 3-phosphate and assimilation of your carbon dioxide. Excuse me. The stoichiometry of the overall path from carbon dioxide to triose phosphate with generation of ribulose 1,5 phosphate. Yeah. This is the summary of the Calvin cycle. Okay. Okay. So as expected, since photosynthetic or since light-induced reaction, yung pinag-usapan nating reaction kanina, it cannot happen in dark. So in dark, the production of ATP and NADPH by photophosphorylation and the incorporation of CO2 intriose phosphate cease. So hindi siya nangyayari. So all except it will only happen in daytime, okay? So what happens in nighttime naman? Meron ba dito? Nakalagay. Ayan. Hindi ko sure kung nailagay ko siya. Pero what happens in nighttime is that doon naman nag-undergo yung different processing of a plant na kung saan hindi niya kailangan ng light reaction. So example is production of uh, cell wall components or the storage. Ayan. All the reaction of the Calvin cycle except those catalyzed by Rubisco, yung cedoheptulose 1,7 bisphosphate and ribulose 5-phosphatase kinase also takes place in animal tissue. Uh, wait lang. Ayan. So that's why yung lacking of these three enzyme in animal cells, hindi kaya ng animal cells mag-fix ng carbon dioxide to produce yung glucose. So yeah, tayo hindi natin kayang mag-produce ng glucose or carbohydrates with the carbon dioxide kasi initially in-excrete natin siya. At the same time, wala tayo ng mga available enzymes sa katawan natin. Specifically yung rubisco. Rubisco is the major component in the introduction of the carbon dioxide from the uh, external source para siya ay makapasok sa cells na sa Calvin cycle. Okay? So yun, lang yan. Wala tayo nito. Rubisco, cedoheptulose 1,7 bisphosphatase, and ribulose 5-phosphate kinase. And questions kayo so far? Sorry may nai-interrupt ako nung itchiness. Questions? Or may clarifications kayo na gustong tanongin? Okay naman so far. Naririnig niyo pa ba ako? Naririnig niyo pa ako? Okay. Okay. Kasi medyo na ulan na dito. Okay. So we have mentioned earlier na ang chloroplast mo cannot always entertain yung mga charged and ionic molecules mo. So may mga specific transporters within the membrane of your chloroplast to allow the entrance and exit of your specific compounds in your chloroplast, in chloroplast. So in this case, this is the light induced production of ATP and ADP. Okay, that will be used up for your Calvin cycle. So I'll explain natin siya in detail. The inner chloroplast membrane is impermeable to most phosphorylated compounds. For example, yung fructose 6-phosphate, glyceraldehyde 6-phosphate, not glyceraldehyde, glucose 6-phosphate, fructose 1,6-bisphosphate. So, hindi siya mapapermit ng mga ganang phosphoryl containing compound. Pero may specific antiporter si chloroplast to exchange the inorganic phosphate with triose phosphate either as dihydroxyacetone phosphate or the 3-PG, okay? Or 3-phosphoglycerine. This antiporter simultaneously moves inorganic phosphate in the chloroplast. Yan. So ito yung itsura niya. Ayan. You have here your light, and within the photosynthesis, you can generate 9 ATP. Ayan, you can generate your ATP. An example lang dito yung 9 ATP kasi yung 9 ATP yung kailangan mo for Calvin cycle. which use up it to produce to be used up to convert your carbon dioxide to glyceraldehyde 3-phosphate. Ayan. And this dihydroxyacetone phosphate that is being used, so it can be converted also to glyceraldehyde 3-phosphate, can enter the chloroplast from cytosol to stroma or from stroma to cytosol. Using this antiporter partial gate in the chloroplast membrane. Okay. So with this antiporter, it now can help in further production of your ATP para mag-undergo yung Calvin cycle mo. Okay, so pag-ulit lang siya diyan. Uh, okay. Just wait. Sucrose synthesis in the cytosol and starch synthesis in the chloroplast are the major pathways which the excess triose phosphate from the pontosis is harvested. Ito yung usually na ano nga na usually na pathway ng iyong triose phosphate in order well, ito yung goal mo naman talaga. Ito yung goal ng plant naman talaga for the carbon dioxide fixation is for it to produce sucrose, the storage of your glucose para sa glycolysis, or starch for longer storage. Okay? Sucrose synthesis releases 4 inorganic phosphate molecules from the 4-triose phosphate required to make sucrose. So sucrose ay siya ay disaccharide of glucose and fructose. So glucose and fructose both have 6 carbon. So meron kang 12. So kailangan, in order for you to form it, kailangan mo ng 3 triose phosphate. Ay, 4 na triose phosphate, sorry. Ayan, kaya sinasabi na 4 yung required to make the sucrose. For every molecule of triose phosphate removed from the chloroplast, 1 inorganic phosphate is transported into chloroplast, providing the 9th inorganic phosphate, which to be used in regenerating the ATP. Ayan, in here. Okay? May kaka-release ka ng isang inorganic phosphate in production of your of sucrose. So yung sinasabi nating inorganic phosphate in here will exit yung cytosol mo to your stroma para naman mag-participate in production of your 9 ATP para mag-undergo ulit yung Calvin cycle. So that's the idea lang, yung sinasabi ng information na to. Okay? If this exchange were blocked, triose phosphate synthesis would quickly deplete the available inorganic phosphate in the chloroplast, slowing ATP synthesis and suppressing assimilation of carbon dioxide into starch. Okay? So yeah, this is how we did ng regulation of the ATP synthesis and carbon dioxide simulation. So as you can see, parang indirectly, yung light has an effect dun sa production of your sugar. So kaya kailangan din talaga ng mga plants ng enough source of sunlight. Okay? The inorganic phosphate triose phosphate antiport system has the indirect effect of moving ATP equivalents and reducing equivalents from the chloroplast to the cytosols. Yan. So, para siyang cycle ulit. So, nangyayari lang dito is that yan. Dihydroxyacetone phosphate form in the stroma is transported to the cytosol, but where it is converted by the glycolytic enzyme, yung 3-phosphoglycerate mo, generating ATP. Okay? So, mag-enter naman siya ulit to your chloroplast, which completes the cycle. So, it is This process helps to regulate yung inorganic phosphate and the formation of the uh, 3-phosphoglycerate and the HAP and production of your ATP and ADP. So, yan. So, this is just the kumbaga indirect effect lang nung shuttle for the formation of and not formation, for the movement of ATP within the system. Okay? Ayan. Enzymes of the Calvin cycle are indirectly activated by light. Ayan. So yung light mo, yung light can help in introduction since are in the induction of your thylakoid and the photosynthetic electron transfer, which as yung pH mo will be lower within the system and yung release of your magnesium 2 plus. Okay? ATP and NADPH concentration increased when chloroplasts are illuminated. As we have seen kanina, na ATP production is assisted by light in the stroma. And dito rin, another micro environment condition that affects yung uh, production of the this or the reaction within the Calvin cycle is pH and magnesium 2 plus. Bakit? Pag marami kang magnesium 2 plus, remember na si Robisco, yung gumagamit ng TPP, ketolase, and transketolase, they are magnesium 2 plus dependent reaction. So, kaya mahalaga na, kaya kapag mataas yung magnesium 2 plus, reactions are more efficient and enzymes are more activated, mas napaparami yung production mo of that glyceraldehyde 3-phosphate and mas maraming napaprocess na carbon dioxide. And imagine that this happens in every second. This is just a 1 unit of Robisco. And given na ang plants, marami silang plant cells at a given area. So for example, sa leaves lang nila, marami silang kayang ma-convert in just a matter of minutes. At the same time, isang leaf lang yon. What if yung buong puno na? Or what if yung buong halaman sa? So you can produce a lot of glyceraldehyde 3-phosphate and and convert yung carbon dioxide to more helpful na organic compound kapag marami kang puno. So kaya naman din talaga ang tinatarget of our environmentalists is magtanim ng mas maraming puno. Kasi it can help us to reduce yung carbon dioxide within the system, within the environment. It is not only for aesthetic or for blocking of areas, pero it will actually help our environment in reduction of the carbon dioxide in our system. Okay? So, at this condition, yung mataas na, yung illuminated na stroma can induce yung pagtaas ng pH. So, magiging more basic siya and increase in magnesium 2 plus concentration. Okay? So, yung affected na enzymes ito is the Robisco and fructose 1,6 bisphosphatase. Okay? Other enzymes that have been indirectly affected by delight are ribose 5-phosphate kinase, fructose 1,6 bisphosphatase, heptulose 1,7 bisphosphatase, and K3P or glyceraldehyde 3-phosphate dehydrogenase sa paanong paraan. Okay, so these compounds or yung these enzymes have disulfide bonds in their system. Okay? So by yung disulfide bonds na yon, they are prone for we call this prone for breaking. And upon breaking these disulfide bonds, it can make your enzyme active. Okay? So kapag close yung or kapag bonded yung disulfide mo, uh, inactive yung enzyme. Kapag break sila, they are active enzymes. Okay? So ano nangyayari sa kanila? Uh, okay? Upon exposure to light, yung photosystem mo, upon reduction, can help in assistance of your disulfide bonds, your thioredoxin. Ayan. When these cysteine residues are bonded, the enzymes are inactive. This is the normal situation in the dark. So kapag kapag walang ilaw, so with illumination, electron flows from the photosystem 1 to ferrodoxin, which passes the electrons to a small soluble disulfide containing protein called thioredoxin. In the reaction catalyzed by ferrodoxin thioredoxin reductase. So si ferrodoxin mo, siya yung carrier of your electron, which assists in the breaking of the disulfide band in your thioredoxin and sorry makes your thyroidoxin having a unbound sulfur disulfide. Okay? So yung thyroidoxin mo, which is now somehow activated, will help to activate naman yung inactive enzymes mo. The same yung sulfur disulfide here sa enzyme mo, mga cysteine residues will be activated upon the donation of the electrons from your thyroidoxin. And kapag ganun nga, within the excuse me, in order for you to produce oxygen. Okay? Yan. And kapag naman sa dark, nagkakaroon ka ng inactivation of your enzymes by creating your sulfide bonds. So, yung nangyari naman dito, si enzyme mo na inactive, wait, in dark, yeah. Yeah, yung enzyme mo na inactive. Tama. Ayun. So, reversible lang din sila in dark. Okay? Okay. Ano to? Okay na. Thank you. Okay. So that's what happens sa ating this is another one. Okay? So that's what happens sa ating Calvin cycle. So we started with carbon dioxide fixed in the organic component ribulose 1,5 bisphosphate in order for it to be converted sa glyceraldehyde triphosphate, which will be used for production of your starch or your sucrose or for energy in form of glucose for the glycolysis and some other parts of the ribulose 1 and some glyceraldehyde triphosphate will be converted naman to regenerate your ribulose 1,5 bisphosphate. So these are just the affected enzymes during the light or during the daytime or during kapag merong source of illumination. So many enzymes sa plant are very light dependent, and this light dependent enzymes are crucial for the synthesis of different biomolecules which is needed by the plants to survive. Okay? Question for the may question kayo for the Calvin cycle? Wala pa. Okay. Lang. Edit lang ako. All right. So this is another type of your um, pathway in carbon fixation. So photorespiration, ano? Ay, hindi pa pala. Yeah. So photorespiration consumes oxygen and produces carbon dioxide. And like photosynthesis is driven by light. Um, so this happens yung, kasi di ba, for the past natin na, na discussion, yung, well, sa Rubisco, it uses carbon dioxide for the carbonylation of your Rubisco. And sometimes, hindi kasi specific si Rubisco for carbon dioxide alone. So it can use up molecular oxygen sa active site niya. And if that happens, nagko-form ka ng different compound bukod kay 3-phosphoglycerate alone. So, kaya siya tinawag na ano yung totoong name ni Rubisco? Siya ay ribulose 1,5-bisphosphate carboxylate and oxygenase. So, hindi lang siya nag-entertain ng carboxyl group. It can also entertain oxygen compound. So, yun yung problem with the Rubisco. Hindi siya specific only for carbon dioxide. So, how does the plant overcome yung ganitong issues niya in help of your in help give it different pathway. Okay? So, due to Rubisco's non-absolute specificity, molecular oxygen competes with carbon dioxide at the active site, and about once in every 3 or 4 turnovers, this leads to formation of 3-phosphoglycerate and 2-phosphoglycolate. Okay? So, yung 2-phosphoglycolate mo is a useless product. Useless siya in application. In order to make it a useful component ng iyong ng plant, it will be converted ulit to 3-phosphoglycerate. So, yung conversion na part na yun is energy consuming, so impractical talaga siya na approach for the I mean impractical siyang gawin, pero well hindi siya may iwasan since once in every 3, so parang 25% or 8% ng oxygen can be uh, oxygenated with the Rubisco. Okay? So yung ribulose 1,5-phosphate mo will have an indiol form. And in presence of your containing your 2 oxygens, and upon addition of your OH group, you'll now have your 3-phosphoglycerate and 2-phosphoglycolate. So this 2-phosphoglycolate is, yeah, metabolically useless product. So what will be the call this? What will be the solution of the plant to use up, or para hindi masayang yung compound na to. So
Oh.
it will be salvaged using the glycolate pathway. Excuse me. This glycolate pathway will convert two molecules of your 2-phosphoglycolate to a molecule of serine, 3 carbons, and a molecule of carbon dioxide. So what happens here is that your mm, just okay. Okay. In your chloroplast, a phosphatase converts your 2-phosphoglycolate to glycolate. So yung 2-phosphoglycolate mo uh, will be converted to glycolate, releasing yung inorganic phosphate mo. So your glycolate here, which is exported to the peroxisome. Ayan. Yung peroxisome mo naman, in the peroxisome, the glycolate is oxidized with molecular oxygen, and the resulting aldehyde undergoes transamination with glycine. So your glycolate will be formed as glyoxylate, and upon addition of your amine group from your serine, you'll now have your glycine. Okay? Sana nakikita niyo siya. Ayan. Pwede pala siyang i-zoom. Okay. Okay? So yeah, yung 2-phosphoglycerate mo, you are using up oxygen here as that will be fixed by your Rubisco, and you will be forming 2-phosphoglycerate. It can be converted in chloroplast by removing your inorganic phosphate. Yung glycolate mo will be transferred to peroxisome, and peroxisome will assist in the introduction of your molecular oxygen again using glycolic acid oxidase to form your glycoxylate. And this glycoxylate will receive amine group from a serine, which is somehow which will be formed then within the cyclic system, cyclic reaction. Okay? Glycine passes from the peroxisome to mitochondrion, and uh, it undergoes oxidative decarboxylation by the glycine decarboxylate complex, similar with the other the decarboxylase that we have encountered sa mga previous lectures natin. So your glycine here will be reduced by your NADH and decarboxylated to release your ammonia and CO2 release in the photorespiration. Now your glycine will be converted to serine. Okay. The glycine decarboxylate complex oxidizes glycine to carbon dioxide and ammonia. Ayan, dito siya. This is what happens to your glycine decarboxylate complex. It oxidizes your glycine to carbon dioxide and ammonia with the concomitant reduction of NAD plus to NADH and transfer of the remaining carbon from glycine to the cofactor tetrahydrofolate. So your glycine here will be mag-remove ka ng dalawang hydrogen, and it is now attached to your tetrahydrofolate, and ayan, it will be decarboxylated, removal of your carbon dioxide. Now you have your here your complex here, and it will now be ammonia will be removed from your compound, and by assistance of your tetrahydrofolate, now yung one carbon containing unit mo will be carried by tetrahydrofolate, which is transferred to your second glycine by serine hydroxymethyl transferase, producing serine. Okay, so yung end product of this decarboxylation process, yung glycine decarboxylase, is the production of your serine. Ayan, which will be which will be okay, which will enter your peroxisome. Yung serine natin kanina, uh, doon nang gagaling yung amine from for the glycine to be for the glycoxylate to be converted to glycine. So yung serine mo will undergo will be converted to hydroxypyruvate, and upon uh, reduction of it, now you have your glycerate. Okay? So, anong meron ulit? So, sa glycerate, papasok siya sa plus stroma mo, which can be converted to 3-phosphoglycerate. Okay? So, that's what happens if oxygen yung i-fix nung diba? Nung plant cell. So, what happens here is just you use up a lot of stuff here. So, you use up energy in here in order for you to attain ulit yung 3-phosphoglycerate, which is what we expected to have for the Calvin cycle. And you use up a lot of enzymes for this. So, yun yung way of plant cell to overcome yung issue ni Robisco na non-specific property niya. Lang. Okay. Okay. The combined activity of the Robisco oxygenase and the glycolic salvage pathway consumes oxygen and produces carbon dioxide. Hence the name photorespiration. And ng glycolic pathway, yung yung yan, yung iyong 2-phosphoglycolate uses up 2 carbon, that is a C2 interaction. Kanina, yung C3 natin, you produce 3-phosphoglycerate, ito glycolate, and may actually inhibit net biomass formation as much as 50%. Okay? Hindi ito nagpo-produce ng energy, so it is an impractical approach talaga. Pero, the plant cannot avoid it kasi large percent will happen pa rin. Okay? So, that's for the C2 reaction. Meron tayong C3 kanina. So, for this naman, yung ating mga C4 plants. Wait, anong oras na? 15 minutes. Okay, tapusin na natin yung kaya natin matapos. Okay, in C4 plants, carbon dioxide fixation and Robisco activity are spatially separated. So C4 plants are those plants that use the process where carbon dioxide is fixed in a 3-carbon product, 3-phosphoglycerate, where CO2 is temporarily fixed into 4-carbon compound. Process. Wait lang. Yeah, they purpose. Where CO2 is temporarily fixed. C4 plants ay yan. Example of these C4 plants ay yung tropical plants, which are maize, sugarcane, and sorghum. So C4 plants traits have to be high photosynthetic rate. So mas mabilis yung photosynthesis nila. So mas mabilis yung production of oxygen and water. High growth rates. So mas mabilis silang tumubo even in the most uh, dry or not most dry, not in tropical. So ito yung country natin. So ito yung mga plants that grow ng mabilis in our ecosystem. Low respiration rates. So hindi siya masyadong affected by the oxygen uh, or the rubisco oxygenase. Low rates of water loss and specialized leaf structure. So may specific leaf structure sila. Mesophyll and bundle shift cells are the cells involved in the photosynthesis in the C4 plants. So this is just the hindi ko sure kung SEM ba to or just microscopic view of the plasmon of the cell wall or plant cell. You can see here daw yung mesophyll cell and the bundle sheet cell. Okay. So bundle sheet cell and mesophyll cell. We will see later ano yung plasmodesmata. So that is an important linkage between the mesophyll cell and bundle sheet cell. Ayan. So ito yung plasmodesmata. Okay? And this is the plasma membrane between the two types of cell, yung mesophyll cell and the bundle sheet cell. So what happens here? In C4 plants, carbon dioxide is fixed to oxalate. So you use a 4-carbon compound. So C4 plants nga, catalyzed by phosphoenol pyruvate carboxylate. So, instead of carbon dioxide, yung ginagamit niya as substrate, it's using bicarbonate or HCO3 negative. So yung carbon dioxide mo, upon addition with water, will give off your, will create bicarbonate, HCO3 negative, and it will be used by the mesophyll cell as the precursor for the production of your sugar or your carbohydrate. So, enter siya ng cell as a as an oxaloacetate. Oxaloacetate is then reduced. Oxaloacetate is then reduced to malate at the expense of NADPH or converted to aspartate by transamination. Okay? Yung aspartate, if ever naman na yung oxaloacetate will be transaminated, may separate pathway for the aspartate to be converted to malate. So lahat ay magpo-form pa rin ng malate. So malate can be transported to your bundle sheet cell via plasmodesmata. So yung malate mo will be oxidized and decarboxylated to yield your pyruvate. Ayan, to yield your pyruvate. So yung pyruvate and carbon dioxide will be produced. Itong carbon dioxide na ito will be now converted by your rubisco to form your 3-phosphoglycerate, then your triose phosphates. The pyruvate, the pyruvate form is converted to phosphoenol pyruvate by an unusual enzymatic reaction catalyzed by PPD or pyruvate phosphate dikinase. Kung bakit siya dikinase? Kasi you are using yung pyruvate phosphate dikinase in two manners. Addition of removal of ATP to removal of 2 units. Removal of 2 units of your of phosphate group to your ATP and attachment of one of phosphate to your pyruvate. So kaya siya dikinase. Yun yung function niya. Okay? So that's what happens sa C4 plants carbon dioxide fixation. So gumagamit ka ng carbon dioxide in form of bicarbonate, and it will enter uh, yung 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 cell by reaction with phosphoenol pyruvate to form your oxaloacetate. Oxaloacetate is for containing carbon, kaya siya tinawag na C4, 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 specific for C4 plants. Or kaya tinawag na C4 plants sila kasi ginagamit nila yung phosphoenol pyruvate uh, to produce yung or to fix yung carbon dioxide. Okay? So yung total fixation pa nung carbon dioxide will happen inside the bundle sheet cell, whereas it will be converted by Rubisco to your triose phosphate. So magsa-cyclic ulit siya in order uh, to continue the production or the fixation of carbon dioxide. Just me now. Okay. So the phosphoenol pyruvate carboxylase of mesophyll cells has a high affinity for bicarbonate and can fix carbon dioxide more efficiently than can Rubisco. Bakit? Kasi sa Rubisco nga, it can either uh, uh, it can either fix oxygen or carbon dioxide, whereas sa enol carboxylase, it can only assist yung or can only entertain yung bicarbonate. Okay? So, ayan, ito pala sinabi pala dito. Unlike Rubisco, it does not use oxygen as an alternative substrate. So, there is no competition between carbon dioxide and oxygen. This is needed nga kasi dun sa plant that can only live dun sa mga tropical uh, setting. Okay? Three enzymes of the C4 pathway are regulated by light, becoming more active in daylight. So these three are the malate dehydrogenase, phosphoenol pyruvate carboxylase, and pyruvate phosphorylated by thyrodoxine dependent reduction mechanism, yung malate dehydrogenase mo. And yung PEP naman is by the phosphorylation of serine residue, and pyruvate is for the phosphorylation. Okay, tapusin natin siguro mga 3 slides pa. Okay? C4, the energy requirement for the C4 plants, it needs 5 ATP molecules for assimilation of 1 molecule of carbon dioxide. So unlike kay Calvin cycle that uses up 9 ATP for the carbon dioxide fixation, kay C4 plants, it uses only 5 ATP. So as the temperature increases, the affinity of Rubisco for carbon dioxide decreases. A point is reached at about 28 to 30 degrees Celsius, at which the gain is sufficiency for and this just explain yung effect ng temperature if ever na kasi plant pa rin yan. So there is a drastic effect pag sobrang pag it's too much naman for it to be exposed in light. Okay? So it can increase yung temperature at yan. Things can happen. Okay. This is another way naman of the plants in how about yung mga plants that live on very hot and very dry environment. So yung mga cactus natin and mga pineapple, how can they thrive in that drastic environment, especially the source or madalang yung source of water. So at sobrang init nung area, especially in daylight. So what happens in them? So meron silang different variation of photosynthetic carbon dioxide fixation. which reduces loss of water vapor through the pores, stomata, by which carbon dioxide and oxygen must enter leaf tissue. So what happens here is that at night, when the air is cooler and moister, the stomata open up to allow the entry of carbon dioxide. So dito siya nag-iipon ng carbon dioxide, which is then fixed into oxaloacetate by phosphoenol pyruvate. Hindi siya kita. Malabo yung copy. Ayan. So yung stomata mo. Yung stomata ng cactus or mga plants that live in very hot places or dry places, it opens up in night. This is this is the advantage of them to capture carbon dioxide. So nag-open up ng carbon dioxide which can be converted by the phosphoenol carboxylate to oxaloacetate. So nag-undergo ulit siya ng C4 plants pa rin ito, and it has been restored to vacuoles. Okay? Uh, the oxaloacetate is reduced to malate and is stored in vacuoles to protect cytosolic and plastid enzymes from acidic environment produced by malic acid dissociation. So, si malic acid, yung malate, can be converted to malic acid, and this conversion only happens inside the vacuole in order to protect yung other components ng cell from a low pH. Remember kanina na we explained na kapag high yung pH, so means basic yung system, it can it can induce yung production of your or fixation of your carbon. Kapag mababa naman yung pH mo, acidic, so it can lower down or it can significantly decrease the activity of fixation. So hindi siya practical for the plant cell. So kaya ini-store niya yung malic acid, an acidic organic compound which can affect yung environment sa vacuum. So what happens next is that during the day, the stomata close up naman, preventing the water loss that would result from high daytime temperature, and the carbon dioxide trapped overnight in malate is released as carbon dioxide by the NADP link malic enzyme. So, ayan. Yung malic acid mo from your vacuole will be released as malate, and it can be converted to pyruvate, glycerate, and phosphoenol pyruvate to storage as starch, or yung carbon dioxide mo will be now undergo to your Calvin cycle. Okay? So that's how this plants, the C4 plants and the CAM plants adapt to their drastic environment. So what happens is this carbon dioxide is now assimilated by the action of Rubisco and the Calvin cycle enzyme. So doon pa lang mag-undergo yung Calvin enzyme cycles kasi daytime. I mean, kailangan pa rin ng light in order for this CAM plants to uh, assimilate or to fix the carbon dioxide. So what happens at night is uh, nagka-capture siya ng carbon dioxide and store it to vacuole, and in daytime, it will close its stomata to uh, fix yung carbon dioxide stored and mag-produce ng energy or ng starting material for its important metabolites and components. Okay? So bakit siya tinawag na CEM plants or CAM plants? Plants that uses resolution acid metabolism. So this is specifically has been observed to a specific plant, and in family of Crassulaceae, and kaya rin siya may acid dito na term kasi it is an acidic here, pero it produce acid component is stored in vacuole and later on converted to an non-acidic component. So kaya siya tinawag na acidic. So that's the meaning of CAM plants, crassulacean acid metabolism plants. So this is specifically for those plants that thrive in dry places and uh, very hot environment. Okay? It's already 5. Hindi tayo umabot. We have, ano pa, we have certain slides pa para naman sa what happens next to your um, 3-phospho glyceraldehyde 3-phosphate. So it can be converted to sucrose and starch, and it can also be converted to uh, polysaccharides like cell wall and other polysaccharide component of your plants. And ayun, so marami pa tayong pag-uusapan sana. Pero yan, it's already 5. Ayan. Uh, Dito lang tayo natapos. So, I will try to record yung lecture for the sucrose and starch formation. Pero sasend ko na to sa inyo later. Sasend ko na ito, yung slides na natapos natin. Ayan. May questions ba kayo or clarification? Okay. How about the others? Wala naman. Okay. So wala na akong question. Actually, doon na nag-end yung lecture natin. Pero baka may questions kayo regarding sa uh, report, sa journal report nyo and written report. So you can ask me or malinaw ba sa inyo yung instruction? Nag? Kailan? Alam. Meron na. Wait lang. Anong name mo? Jericho. Okay. Sorry, hindi ko siya nakita. Sige, re-replyan kita later. Okay. Ayan. Tapos, tip ko pala sa inyo regarding sa topic nyo. So, better na mag-communicate kayo with other classmates nyo para ma-avoid yung repetition ng topic. So, yun lang naman. Pwede naman kayo mag-consult pa rin sa akin regarding your topics. Tapos, tama ba? Kailan na binigay ko sa inyo yung date for submission of abstract? July 4 ba yun? 4. Okay, sige. Ayan, yun lang naman. Okay, so kung wala na kayong question, we can end na our discussion. Thank you, everyone.